Thanks all for logging in, and it's our great pleasure to welcome Professor Alix Deymier to the MateriAlZ, or however you pronounce it, MateriAlZ seminar series. Dr. Alix Deymier is actually one of our first speakers on anything related to bio, so we are really excited about it. Last year, when we started the series, we didn't have too much of a bio focus, and we got a lot of pushback on, no, we need bio, and who better than to turn to Alix to educate us on biomaterials. Alix is an alum of MSc in, not math sci, MSc at the UFA. She graduated in 2006, and she was, I mean, I know her because her, even if everyone forgets her names on the Chapman Fellowship thing on our board, right?
I'm on the wall.
You're on the wall. Moved to Northwestern, WashU, Columbia, and now she's a professor at University of Connecticut. So the floor is yours, Alix. We are really excited to hear your seminar.
Thank you. Thank you, everyone, for listening to this talk. I'm very excited to give it. It's really it's a brand new talk, so it's exciting to show you some of the stuff we've been doing, especially during the pandemic. So in case anyone didn't get the joke, here we go. The amino acid, it's amino acid. But the worry really is that acids are bad for you. And I didn't start off in acid. That wasn't, bones were not my thing. Back when I was at Arizona, I was on the cover of Arizona Engineering in 2006. And I used to work in Dr. Vandiver's lab doing ancient ceramic reconstruction. So my materials has always been focused on ceramics, but it used to be art ceramics. And we have since moved on to biology and looking at ceramics in the context of bone. So I want you to visualize yourself as we start. It's Labor Day, you're outside sitting by the pool, trying not to get bitten by mosquitoes, and you're drinking a soda. Raise your hand if some of you imagine that you'll be doing this weekend. Yeah, we got a couple people, fabulous. Now, maybe some of you are drinking Coke. And that's fine, except you might not know that the pH of Coca-Cola is about 2.7. And that is as acidic as stomach acid. So every time you're drinking a Coke, you're putting in a lot of acid into your body. And so some of you who are maybe Pepsi drinkers, do we have some preferred Pepsi drinkers over Coke? Well, you're doing even worse because the pH of Pepsi is actually 2.53. So every time you're drinking a soda, you're actually putting in a really strong amount of acid into your body rather quickly. Now let's talk about what happens when you drink soda. You start off your day, your pH is 7.4, which is physiologically stable. You're happy, everything is going well. But once you drink that soda, that acid is so strong that you actually see a decrease in pH in the body. If I were to measure your blood after drinking soda, you would have a reduced pH. And your body hates having modifications in pH. It's actually extremely sensitive. Even a drop to like 7.35, so only 0.05 change in pH units, dramatically increases your chance of mortality. So body's like, nah, we do not want this change in pH. So what it does is it recruits the acid hero, which is actually the kidney. So in normal healthy people, you drink your soda, your kidneys kick into high gear, and it eliminates the acid through two methods. Either it takes the acid and it puts it into your urine and you're just gonna have acidic urine, or more importantly for our talk today, your kidneys also regulate your bicarbonate metabolism. So bicarbonate gets released out from the kidneys into the blood, and that is what's actually going to neutralize a lot of that acid. So if you are a healthy young person, you're going to be just fine because your kidneys are going to kick in. But there are very large swaths of the population that do not have functioning kidneys. That could be because they have something like chronic kidney disease, which affects about 15% of the US population, so a huge percentage. Or maybe it's just that you're eating a diet that has so much acid that you can't have the kidneys do all of the work. They just can't do enough. And that's true of pretty much anyone who's on the keto diet. The whole goal of the keto diet is to hit acidosis. So when you're doing something that affects your kidneys or have a condition that affects your kidneys, you need a new hero to come along to regulate your metabolism and your pH, and that is the skeleton. Now I know it seems strange, like you're like, okay, I get it, kidneys filter things, but how in the world is the skeleton going to have an effect on the acidity of my body? Well, to really understand that, we have to look into what bones are actually made of. So the bone, it's really just a three-phase composite. On one hand, you have this compliant protein. About 30% of your bone is made of collagen. That gives it a lot of toughness, a lot of ability to like arrest cracks. On the other hand, about 60% is mineral. And about 10% is fluid, and that's going to be blood, synovial fluid, and other body fluids. Now, what's really helping in terms of acid-base balance is the mineral. That's really what we're looking at in the case of these studies. So mineral in bone, you're like, okay, great, what is it? Well, for years, people said, oh, it's hydroxyapatite. Hydroxyapatite is a geological mineral. It makes these really big centimeter-sized green crystals with beautiful hexagonal faceting. It's a calcium phosphate. But the fact is, that is not what bones are made of. This is a lie. We now have so much more information that says that what most bones are made of in most mammals is a carbonated apatite. So this is an apatite, it's still a calcium phosphate. But instead of just being calcium phosphate and OH groups, we now have the substitution of additional carbonates, and we have the substitution of sodium. Now, maybe you're dinging right here and you're like, carbonate. We already learned that bicarbonate is really useful for neutralizing acid. So this must be important, and you would be correct. But the carbonate has tons of other effects on the mineral. So if we think about bone, it has about 6 weight % carbonate. So not a ton, but pretty significant. And that 6 weight % carbonate means that we go from having a centimeter-sized hexagonal crystal to suddenly having nanometer-sized crystals that are about 50 by 30 by 4 nanometers that have this kind of platelet shape. On top of changing from big crystals to small crystals, we decrease the crystallinity and we increase the solubility. So biologically, by adding carbonate, we've actually have smaller crystals that are less crystalline and more easily dissolved. Well, that seems like it's perfectly handy to serve as a way to serve as a carbonate manufacturer. So these crystals seem to be tuned biologically to be carbonate releasers. Now that's a lot of material science, great, but what happens clinically? Well, clinically, we know that if you have a bone in your body, which we all do, you have your skeleton, when it comes into contact with acids, it releases bicarbonates and helps to neutralize the acid. And we actually see a lot of evidence that the skeleton helps to regulate the acid effectively and can bring people back into homeostasis. So you're thinking, great, fabulous. The problem is, in order to release that bicarbonate, you have to dissolve your bones. And that means that you go from having a normal bone structure to an osteoporotic bone structure. You have decreased bone mineral density and you get an increased fracture risk. So even though it's great in terms of bringing your pH back up, it's really bad in terms of bone health. Now the question here isn't, does this happen? We do know that it dissolves and that it does all these things. The question is, how does it happen? Now, the dissolution mechanism of bone has been hotly debated for a very long time. Seems like such a simple question. And there's really two main camps. On one side, you have the physiochemical dissolution people. They say you'd have acid in the body, the bone comes into contact with this acid, and essentially you're just dissolving these crystals which are primed for dissolution because of their composition. On the other side, you have the cell-mediated dissolution people. And they say, well, no, it's not just physiochemical. our bones are full of cells called osteoclasts, which resorb bone and release those ions out into the body. So they say it's all about the cells. And there is quite a bit of evidence that says maybe it is the cells. And biologists are just gung-ho about the cells. They don't believe in any physiochemical. It's just really been cell-focused. And the reason they're so motivated is because they've done experiments where they take these osteoclasts, these bone-resorbing cells, and they add acid to it in a petri dish type of environment, so in vitro. And when they do that, they see that the osteoclasts replicate at higher rates and they become more active. So they said, great, if I add acid to a cell in a petri dish, it multiplies, it becomes more active. So it must be the cells. That's what's controlling the dissolution mechanism. However, there's a ton of evidence that in vitro studies with cells in petri dishes do not recapitulate what actually happens in the body. The body is so much more complex. So one of my goals in the study I'm going to show you today is to try to understand which mechanism is the primary mechanism, if they have changes with time, and which one is really occurring in terms of a clinical relevance. And the true thing here is that depending on the mechanism, you would treat it differently. So if I just have acid that's coming in and dissolving my bones, I'm just going to give my patient Tums. This is literally our treatment, is we just fill people with carbonates. Give them Tums, hopefully it brings down their acidosis. You've eliminated the acid stimulus, and therefore you don't have dissolution. That would be the fastest way to treat it if the physiochemical response is really the cause of dissolution. But if it's the cells, you'll probably want to give them a bisphosphonate. A bisphosphonate is just a drug that stops osteoclasts from dissolving bone. If you do that, you should be fine. You can treat the acid later, but you can immediately stop the skeletal response. So we really need to understand which of these mechanisms is occurring, which one is causing the worst consequences, and that will inform how we treat people in the future. So in my lab, in order to differentiate these different treatments, we've used kind of a multi-scale examination technique. So on one hand, we're going to work with a full mouse that has acidosis, that's completely alive, and we're going to try to understand what affects the bones. On the other hand, we're going to do an ex vivo study where we take the bones out of healthy mice and put them in acid and try to see what happens there. And finally, we're going to go in vitro, where we're going to eliminate the whole complexities of bone with its multi-phase characteristics and just examine how it works at the crystal level and its dissolution. So let's get started with the in vivo. So the way that we induce acidosis in mice is by giving them a weak acid. So essentially we make an artificial soda. We replace all of their water with low doses of ammonium chloride. So you can see here's my acidosis mice. From here on out, in this section, red will be acidosis and green will be control. So in my acidosis mouse, it's fine for 7 days, and then I give it increasing doses of acid in its water. So it's drinking artificial soda. My control mouse, it's just getting normal water. It's just chilling in the cage, no changes. I'm going to collect urine information to understand how the kidneys work. And I'm going to get blood information to understand what the pH of the blood might be at that point. Now, I would like to point out that the pH of our ammonium chloride is significantly higher than any soda that we would be drinking as humans. It is actually illegal for me to give Coca-Cola to mice because the pH is too low. Just something to keep in mind. So what happens when I do this? Here are the results from the pH from the blood and the urine. So here's pH as a function of time. And then the dotted lines up here are blood. So green is control. You can see, okay, pH didn't really change. But in the red, you can see that in my acidosis group, once I induce acidosis with this line, I have a drop in blood pH 1 day after they start to drink low doses of soda. After 14 days, we've actually maintained that low blood pH. In the urine, same thing. It immediately drops off after we add the ammonium chloride, and it stays low. So we've successfully induced acidosis in all of these mice. Now, what does that mean? If we keep looking at the blood, we see that at one day, the calcium levels in the blood are pretty much unchanged. But at 14 days, so 14 days of drinking soda, the calcium level in the blood is significantly higher than in my control mice. So what this tells me is that high calcium is probably coming from my bones. I probably have some level of calcium being extracted from the bone and put actually into the blood supply. So I know I've induced acidosis, and now I know that I probably have bone dissolution as indicated by the increase in calcium in the blood. Okay. If I'm seeing that there's probably dissolution, there's probably going to be changes in the composition of the bone itself. So to get compositional information, I'm actually going to go ahead and do Raman spectroscopy. And Raman's going to give us a whole slew of information, but I'm going to break it down into two main factors. The first factor that we're going to look at is the mineral to matrix. And that's telling me how much mineral I have in my bone compared to the amount of collagen in my bone. The other factor we'll look at is the carbonate to phosphate ratio. So I told you the bone mineral is a calcium phosphate that has carbonate in it. So by measuring that carbonate to phosphate ratio, I can determine if there's changes in the amount of carbonate within the bone crystals itself. What you can see is that my mineral to matrix on the bone exterior is unchanged. I might have some dissolution, but it's pretty minor. I'm not really dissolving large amounts of crystals. However, at day 14, I have significantly reduced the amount of carbonate that is present in the bone mineral. So essentially, I've known that calcium is being taken out of the bone, but I'm also seeing that the carbonate is taken out of the bone. Now this is good, right? We want the carbonate to come out because that buffers the pH. Okay, great. That seems to make sense. I take my bone, it's surrounded by acid, some of it dissolves, it takes out carbonate, it buffers, everything's going well. Now, what happens in the interior of the bone, right? This exterior is exposed to body fluids, it's fine, but the bone is dense. So what happens on the inside of the bone? And that's where we get our first surprise. It completely changes the game. Here we see that the mineral to matrix actually decreases. So now, instead of having a situation where I'm not losing a lot of mineral, I actually have significantly less mineral on the inside of my bone than I do on the outside. Bicarbonate, on the other hand, is unchanged. So what this points to is the fact that I must have two kind of different mechanisms. On the outside, maybe I'm exposed to body fluids, I'm getting some sort of physiochemical response. And on the dense interior of the bone, maybe this is a cellular response where I'm getting changes in the bone turnover and it's putting in less mineral, but not changing the carbonate. At least this is going to be our hypothesis for now. So if indeed my cells are doing things, which this seems to suggest, and I'm getting some sort of physiochemical response, which my exterior measurements seem to suggest, maybe I'm going to get a change in the shape and size of my bone. So the first thing we did after Raman was to go ahead and do micro-CT, which is microcomputed tomography, and essentially gives us cross-sections or 3D images of our bone structure. And what we ended up finding was that having acidosis, so 14 days of soda drinking, caused a significant change in the shape of the bone. They were much more compact, smaller, and they had a lower moment of inertia. So 14 days, I'm getting all these chemical changes, but these cells must also be reworking the organization of my bone. So I'm thinking, man, like maybe the cells are a pretty big deal here, right? If I have a change in composition and I have a change in shape, then it's pretty likely that I'm going to have a change in mechanics. So we take these same bones and we ended up doing 3-point bend. Now, what we find in the 3-point bend results is that we don't have a lot of changes in things like stiffness and strength, but we do see an increase in resilience and an increase in fracture toughness. Now, this may seem surprising because I told you earlier we're going to get increased fracture clinically, but the early stages of acidosis effects on bone actually does show an increase in toughness followed by an increase in fracture. So maybe this is indicative of early acidosis response. It is only 14 days. Now you may be thinking, okay, you had your change in size, you had your change in composition, like what's causing that toughness to go up? Well, our theory at this point is that your initial bone had a certain mineral composition and a certain amount of mineral. But remember, we saw that the inside of the bone actually had a reduction in mineral to matrix. That means that our composite, you're taking out the stiff reinforcement of ceramic and you're leaving behind a much more polymeric proteinaceous structure. So if I'm having a crack, whoo, come back. Oh, presentation, come back. If I have a crack propagating through that material, it is going to encounter a much more plastic material, which can have the formation of a plastic zone at the crack tip, and therefore you're less likely to fail. So this is our theory at this point. We're getting both physiochemical and cellular, we're getting changes in composition, and that is going to affect my toughness by arresting cracks through the formation of a plastic zone. Now I am seeing these changes that are probably cellular. You're not going to dissolve bone into a new shape. That seems to really be a cellular behavior. So I collected the cells from these bones and I did RNA analysis. And RNA analysis, what it tells us is what genes are being activated within the cells that are present in bone. So here you can see I have fold expression change. So that says for a gene of interest, how many times higher or lower is the expression than in your control group? So if it's equal to 1, it essentially means that the expression of that gene is identical to what you would have in a control group, no change. On the x-axis here, we have the 4 genes of interest. So let me give you some primer here because we're not all bone specialists. So the first gene we looked at was ALP. ALP is created by osteoblasts, which are cells that grow bone. So if your ALP is increased, you would say, great, osteoblasts are active, they're making more bone. So ALP making bone. Over here on the far end, we have TRAP. TRAP is made by osteoclasts, which resorb bone. So if you have high amounts of TRAP, you're usually like, oh, we're in bone resorption. We're definitely eating away bone and releasing those ions. So TRAP, that's dissolution. OPG and RANKL are just a little bit more complicated. They're both made by the osteoblast. But what RANKL does is it activates osteoclasts. Essentially, the osteoblast is saying, oh, I probably made a little bit too much bone. Hey, osteoclast, can you come over and take it away? So a high level of RANKL usually indicates resorption. OPG, it inhibits RANKL. So if the osteoblast changes in mind, it's like, oh, make more OPG, stop the RANKL from getting to the osteoclast. So it's associated with bone growth. So these two are associated with bone growth. You can see they're about equal to 1. So that doesn't seem to be causing any changes. And here, these two are associated with bone removal. And even then, there's no significant difference compared to controls. Now, these were taken at day 14. And I'm thinking, man, I have all these changes to my bone, and yet it's telling me that nothing has changed in my cellular activity. That seems really, really fishy. So I say, okay, well, maybe there's something with the TRAP. Maybe it's time dependent. So what we do now is we went ahead and we took bones from day 1, 3, 7, 14, and controls. And we stained them using a stain that stains for osteoclasts, so bone resorbing cells. So you can see these little purple dots here. Those are osteoclasts. We counted how many of those we had per area of bone. That's what we have here. And what you see is that indeed, between control and day 14, there's no change. Whew, my RNA data makes sense. We got it. But at day 3, I have an increase. So if you remember back to my intro, I said that in the Petri dish, you add acid and you get more osteoclasts. Well, that only seems to be true for a short period of time. And most Petri dish experiments cannot last an extended period of time. So maybe what all those biologists have been seeing is just a snapshot of what's actually happening in the body. So all those changes in the mineral composition and the organization of the mineral to matrix ratio and the structure of the bone, it's happening really fast. All happening in the middle here, and by day 14, you're already back to stable. So this was really interesting that we had this time dependence in bone resorption. So we thought, well, do we have it in bone formation? Nope. Bone formation is completely unaffected. So now we're starting to really get an understanding of how bone structure and material properties are probably playing a pretty important role, because the cell effects seem to be relatively temporary. So let's really recap this section just to make sure we have it all down. We have our bone. You drink your soda. The first thing it does, calcium and carbonate. They come out, that carbonate turns into bicarbonate. It helps to buffer your body. Happy, we're good. Now that process does seem to stimulate some cell behavior, which causes your body to essentially start changing the way your bones are shaped. So you have a decrease in the size of the bone and the organization of the bone. And all these things together cause it to change in its mechanical properties. You see that up in toughness. Now that acid is also affecting the cells, and that it is causing those osteoclasts to replicate and be more common, but it appears that that's super time dependent. Now what you can see from this conclusion about the in vivo is that we really haven't separated out anything. The body is so complicated that we can't possibly isolate physiochemical responses from cellular responses in a mouse. It's just really complicated. So we decided that the next thing we needed to do was essentially eliminate the cells. And to do that, we switched to an ex vivo model. So here we use the same mice, but instead of using the ones that had the acid, I'm taking my control mice. I don't want to kill any more mice, so instead of using their femurs, I'm going to use their tibia, fibula, ulna, and radius. So I take those bones out of my control mice, and I freeze them to make sure that all of the cells are dead. Now this is purely a materials question. I have not had any sort of biological response from here on out. Cells are gone. Good. Now I'm going to take these bones and put them into solutions of varying acids. And that's going to tell me what happens to these types of bones in acidic situations. So what are my conditions? This is important. Well, first of all, I'm going to do just bones. I need to have some sort of baseline. This is the composition, structure, and mechanics of the bones before any sort of acid exposure. Then I have just my solutions. The solution we selected is PBS, also known as phosphate buffered saline. It's a solution that represents body fluids in many ways, but it has lower levels of calcium and it's not super saturated. So it's not going to precipitate its own crystals, which is actually very important in these questions. We then pH that PBS to pH 7.4, which is physiologically stable. pH 7, which is the acidosis in a mouse. They can handle a lot more acid. And then pH 5.5, which is super extreme, but is actually what your teeth feel in your mouth environment on a super regular basis. We then take bones and put them in each of these solutions. So we'll be able to know how exposure to these acid body fluids actually affects the bone themselves with no cellular behavior or exposure. We left them in the solutions for 5 days. Now, it's a lot of detail, don't worry. It'll be much easier as we keep going. So first we wanted to know, if I just put a bone in a solution, will it buffer? Does it have that ability to actually buffer the pH? What you can see is that the answer is yes. So here's pH, and down here is the time. So we receive up to 5 days, but we took pH at multiple time points. And you can see at low pH shown in red, here's my control group. So this had no bones in it. Now the pH essentially stays at 5.5. But if I have bones, man, they are so good at buffering. I was able to raise the pH by almost a full pH value. Even at 7, after about 3 days, I start to see significant changes where we are successfully buffering and raising the pH beyond 7. At 7.4, it's not really doing much, but that's kind of normal because there's bones are used to being in 7.4. So, phew, okay, all is well. But we can confirm that indeed, when you put a bone into a solution of an acidic body fluid, pseudo body fluid, you are releasing some sort of buffering moiety. So great, this is representative of in vivo, very good. What a surprise. Just like the in vivo system, we're also releasing calcium. So the behavior does seem to be very similar. And you can see that the amount of calcium that was leached out of the bones and into the solution is correlated to the pH. So as I have a lower pH, more calcium is being taken out of the solution, of the bone and into the solution. So we're going to do the same thing. Let's go ahead and do some Raman and see what happens. Here's my exterior Raman. As you can see, my mineral to matrix ratio doesn't change, just like what I saw in my in vivo system. Taking a bone, putting in an acid, even at 5.5, I do not see a change in mineral to matrix ratio on the bone surface. I do, however, see a decrease in carbonate. This is behaving exactly like my in vivo system. Decreased carbonate, no change in mineral to matrix. What about the interior of the bone? Oh, let me give you that visual. On the interior, in the in vivo system, we saw that the mineral to matrix went down significantly. Here we see no change. So we cannot recapitulate the change on the bone interior with just putting dead bones in acid. Similarly, the CO3PO4 doesn't change. So by comparing the in vivo data to this ex vivo data, we start to see that maybe cells are responsible for what happens on the interior of the bone, but not for what happens on the exterior of the bone. This interior shift may be caused by this bone remodeling shift that happens around day 3. So interesting. Now, we did say that the cells were probably responsible for the change in shape and size. And indeed, if we look at moment of inertia and average centroid, nothing has changed if I have no cells. So this does confirm that the cells are probably responsible for the change in shape and size. But now, here's where things start to get really kind of interesting. When I look at the mechanics, the toughness of my material, of my bone itself, as a function of pH, acid exposure increases the toughness. So with no cells, I'm actually able to recapitulate what I saw in vivo. So no cells, just bones in acid, I can recapitulate the mechanical change that I saw in vivo in the mouse that had acidosis. Now this is a bit of a confusing thing because I had theorized that the reason that the toughness went up was that the interior was modified, that I had a decrease in mineral to matrix ratio, which made a plastic zone. But I know that in this system, the interior didn't change. I didn't have this loss of mineral to matrix ratio. So it really becomes a question of like, what is happening here? Is this not what I expected? So I had to think back to all the other things that were occurring here. I had this change in composition, the carbonate was taken out. So maybe the mechanics isn't a result of mineral loss, but it's caused by a change in the mineral itself. Maybe. And that's why we went to the next step, which is looking in vitro at the mineral and how it behaves. So isolating mineral from the bone is very difficult. You cannot get rid of that collagen without really affecting the composition of bone mineral. So what we do instead is we make our own biomimetic carbonated apatite. You just take a carbonate containing moiety, a phosphate containing moiety, and a calcium containing moiety, and you mix them in an aqueous environment at temperatures close to body temperature. If you do that, you can actually precipitate crystals that are nanocrystals with composition, structure, and lattice, so atomic lattice, that looks almost identical to what you have in bone. So we can replicate bone crystals in the lab. We do this all the time. So we went ahead and we did this, and we made biomimetic crystals that had about 6% carbonate. That's what we see in bone. It recapitulates the right type of size, so we're good to go. We then took this powder and we put it once again in PBS solution. Here we did pH 8, 7.4, and 5.5 to get a basic time point as well. We left them in the solution for 3 days, and then we go back and we try to analyze everything. So we see that when you just put crystals of carbonated apatite into solution, you do see a buffering ability. So the pH, if it started at 8, it actually goes up a little bit. 7.4 goes up a little bit. 5.5, it goes up a lot. So the same thing we saw with the ex vivo bone, we see here if we just put mineral into the solution. So probably releasing some sort of buffering moiety like carbonate. We also looked at the solution and saw that calcium was also released. So seemingly this setup is working. It's mimicking the ex vivo system. I do not know why it's lower at 7.4 than 8, one of the great mysteries. We will do this data again. But it does appear that we are releasing the same types of ions that we would release if you had a bone in solution. So, okay, we're good. Now, if we do Raman, here we're doing Raman, here's the change in carbonate to phosphate. We see that just like in the bone, we have a decrease in carbonate. So the carbonate is lost, and the amount of carbonate that's lost is dependent on the pH. So once again, we seem to be doing all the right things. These crystals are appropriate. They're behaving in the same manner. Good to go. But now we have the advantage that we're just looking at a powder of nano mineral, essentially, which gives us the opportunity to much more easily do other material science analyses. And the one we were most pumped to do, which is my favorite technique, is X-ray diffraction. So in X-ray diffraction, we can get a change in lattice spacing as a function of the pH. Down here in the solid bars, this represents the 002 lattice spacing. If we look at the unit cell of hydroxyapatite or carbonated apatite, that represents this long axis, which is the C-axis. And what you can see is after acid, we actually have a decrease or a compression of the C-axis. We also see that this, the 310, which is the A-axis or the horizontal axis, has an expansion. So just by putting these crystals in slightly acidic solutions for 3 days, we see that the lattice spacing has changed as well as the composition of these crystals. Now, this may seem a little esoteric, like how does this relate back to my clinical question? Why am I even looking at this? Well, the key is if I know that I have carbonate loss and I know what the change in lattice spacing is, I can actually get information about what type of carbonate I had in my system. So here, you need to learn this now. Carbonate can go into carbonated apatite in 2 separate ways. The first is A-type carbonate, and that's when carbonate substitutes for OH groups. So you're putting in a big moiety instead of a little guy. The other is B-type carbonate, and that's when it kicks out a phosphate, so a big ionic group, and replaces it with the carbonate, which is a smaller ionic group. Now, obviously, if I'm taking something small out and putting in something big, or taking out something big and putting in something smaller, those are not going to affect the lattice in the same way. So if we lose A-type carbonate, I would expect an expansion of the C-axis and a contraction of the A-axis, which is not what I see. But if I had a loss of B-type carbonate, I should see a contraction of the C-axis and an expansion of the A-axis. So ding, 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 we have lost B-type carbonate. So we know that by putting these types of crystals into acidic solutions, we're actually removing carbonate specifically, not from the surface of these crystals, not from the A location of these crystals, but from the B location of these crystals. Now, again, why is this important? Well, because we've done a lot of research on the effect of B-type carbonate on crystals. We know that if you decrease your B-type carbonate, that will tend to mean that you have an increase in the size of the crystals, that you have a decrease in the solubility of the crystals, and that you have an increase in the crystallinity of the crystals. So we thought, maybe if that loss of B-type carbonate is causing all of these changes to the structure of my crystal, it could be affecting the mechanics. And to understand if that mechanics changes, we had to develop a really complex mechanism. As many of you must know, measuring the elastic modulus of a nano ceramic crystal is not easy, but we did find a way to do it. So what we did is that we went to the DAISY synchrotron particle accelerator that causes high, essentially lasers, so monochromatic beams of X-ray. We went to Hamburg to do this. And what they have is they have a pressure chamber, which is X-ray transparent. So we took crystals with 5 different types of carbonate, so B-type, but different levels. And we pressurized them by putting them in PBS and essentially cranking down the pressure. Like we had to manually crank down the pressure. So we're applying a stress through hydrostatic pressure. We're then sending X-rays through the crystals themselves and getting an X-ray diffraction pattern. Now this X-ray diffraction pattern, just like you would do in the lab, gives you information about the lattice spacing. Now, because I can see what the lattice spacing is at each different stress or at each different pressure, I all of a sudden have a strain measurement. So by doing this experiment on these crystals with different levels of carbonate, what I can get is a stress strain diagram. So strain, stress, and because I can get it for the 002 and the 310, I can actually get the strain for both the C-axis and the A-axis. From this bunch of math, you can check out the paper, we can get essentially the elastic modulus along the C-axis and the A-axis, because these crystals are anisotropic, as a function of carbonate content. And what we see is that unlike on my possible theory that the carbonate might be making these softer and therefore more likely to arrest cracks, the loss of carbonate actually leads to an increase in modulus. So you can see as I go to lower and lower carbonate levels, I have a higher and higher modulus, both along the C-axis and along the A-axis. So if my crystals are getting stiffer, that means my composite is getting stiffer, which means it's likely getting less plastic. And that doesn't explain the crack arrest. So why is my toughness going up if my crystals are getting stronger? And this is a question that we're still working on. Possible theories is that there is mineral loss, but that our Raman techniques that we're using now are not accurate enough to measure that small mineral loss, and that it may actually be enough to cause a change. Another is that maybe the collagen is being modified. When you work with composites, it's so easy to completely ignore one of the phases. But the collagen could be cross-linking or losing crosslinks. It could become denatured. Denatured collagen is jello. That would mean turning something that's got the stiffness and toughness of skin into jello, which would be mechanically very affecting. Or maybe it's something to do with the microstructure. We do have pores filled with fluid, right? 10% fluid. The size and orientation of those pores at the micro scale, which we didn't measure, might be modified, which could create little stress concentrations or little tortuosity pathways, which could limit the crack propagation. So overall, what we found by looking at all 3 of those levels is that when acid interacts with bone, we have release of calcium and carbonate. The carbonate level within the bone goes down. Your mineral crystals get stiffer. But if you have cells, you're also getting less mineral on the interior. We know that acidosis does promote the formation of cells. However, that formation of cells seems to be temporary. we don't see a lot of long-term changes to that. That behavior of the cells doesn't appear to be responsible for the changes in mechanics. When we put this in acid, that's enough, the toughness goes up. So even though biologists love to tell you that the cells are the key factor in bone changes during acidosis, the mechanical behavior, which seems to be honestly the most important, isn't caused by the cells. It seems to be regulated by the physiochemical mechanism. So instead of giving bisphosphonates to people with acidosis, indeed, we should just be giving them Tums. Bringing down that acid, getting it so that the physiochemical dissolution stops. That seems to be the best way to stop these mechanical changes. So there's obviously plenty more to do, but we've really learned a lot over the last 4 years. My lab's only been here for 4 years, about how this bone interacts at the physiochemical and cellular level. I'd like to acknowledge so many people who worked on these projects. Anna Peterson, a graduate student who just graduated last month. She did all of the in vivo work alongside with my graduate student, Michaela Moody. Margaret Eason, who is an amazing undergraduate student who did all of the ex vivo work. And then Stephanie Wong and Michaela Moynihan, who did all the work for the in vitro data. Also the people at the Daisy Synchrotron, people at UConn who helped with mechanics, who helped with RNA, who helped with histology. And then my funding, NSF Career Grant, which came last year, which was very exciting. And then the Helmholtz Association, which funds all of our Synchrotron work. So, so fun to tell you guys this very exciting story. And I see that there's questions in the chat already, which is very exciting, but I'm happy to answer any questions that you might have.
Thanks, Alix. This is great. Open up the floor to the questions, and then people try to, try to type in your questions, or at least raise your hand or something, so that we can then curate and get people to ask questions in order. Is that fair, Ankit?
Sounds good.
Okay, I think Natalie, you should probably start off the questions.
Okay, so I see, Natalie, if you want to speak, that's fine. I can also, I see your question here.
I was going to say, can you hear me?
Yes, I can hear you.
Okay, I know I've been having issues with my wireless headphones. So, yeah, so I know that you had seemed to focus on people that already have health problems, particularly with their kidneys. They're more susceptible to this bone degradation. But what about somebody who is like young and healthy? I know my husband, he drinks a lot of soda, and I have been trying to convince him to decrease his soda intake.
Yeah, I mean, this is the question I get every single time I talk on this subject. And the fact is, it really is a question of balance. If you are drinking so much soda that your kidneys cannot keep up, right? These mice had no kidney disease. Their kidneys are totally fine. But we just gave them so much soda that the kidneys couldn't handle it anymore. And in that case, you're going to get bone dissolution. There's really no way around it. So decreasing soda intake is always a good thing. The other big source of acid is protein. So if you eat protein, you are turning all of that protein into acid. And that's why people who do keto, acidic. People who are like meat and cheese people, you do the blood tests on them, they're going to be acidic. So it's not just soda, it's actually all sorts of diet intakes: meat, cheese, soda, acidic drinks, coffee, also acidic. People start looking up all the alcohols, almost all of them are acidic. I think gin is the only one we found that isn't quite acidic. So yeah, in the end, this data does seem to indicate that making sure that you're eating basic foods, which of course are things like leafy greens and vegetables, is good for you, and eating too much meat, cheese, and soda is bad for you. Nothing new there, sorry.
Thank you.
Of course. Krishna's like, I'm already vegetarian, I've got this.
Yeah, I'm so glad. In fact, I was just texting Elsa. that she needs to give up everything. Sorry, Elsa.
I know. I told him I had to give. I have to give up soda now, and maybe meat too.
I know, it's terrible. And it's so many people do keto. You see, like, keto diets are so popular and they do work well for weight loss, and cardiac wise they seem to be quite good because of the way they stimulate the cardiac tissues, but it's just terrible for your skeletal system. So I see Pierluca has a question.
Yes, thanks. Hi, Alix.
Hi.
Thanks for the very enlightening talk, and thanks for confirming that my gin and tonic diet is actually good for me.
There you go.
So my question was, you mentioned that you make these artificial crystals in your lab because you cannot remove them from the actual bone, or you cannot dissolve the collagen without actually denaturing the crystal.
Yeah.
So my question is, how do they actually know what's really in the bone?
So the reason we know what's in the bone is mostly from non-isolated studies. So if we do things like Raman, for example, we can do Raman on whole bone and still get information specifically on what's happening in the crystal from the phosphate, the carbonate, those types of signals. Yeah, isolating them is almost impossible. Actually, the professor who works 2 doors down, she tried for her graduate studies and she was like, it's a nightmare, don't ever try it. So doing whole bone analyses and isolating them, we do X-ray diffraction on whole bone, which also just gives us crystal information. So things like that. So doing whole bone measurements with techniques that don't require you to take out the collagen, that's how we get the composition. So Raman, FGIR, X-ray diffraction can essentially allow us to get information while ignoring the collagen.
Is it something you would maybe do, or do you think it would give you more realistic results?
I mean, when we do it on the bones, that's essentially what we're doing. And we do have, with the bones, it's a little tricky because once you start crushing them, once you start modifying the bone, it gets harder. So doing X-ray diffraction, which usually requires a powder, is very difficult on whole bone. But we have done lots of synchrotron on whole bone where we can get lattice structure. And we have a proposal into Argonne National Lab to do that, to do whole bone X-ray diffraction, actually, hopefully X-ray diffraction tomography, so we could get it as a function of position as well. So fingers crossed that that gets in. But yes, that would give us really accurate spatial resolution of changes in mineral composition and lattice.
Thank you.
Yeah, of course.
Alix,
Can I
yeah, go ahead. Yes, of course. Go ahead.
Hi, Alix. It's very nice talk and really enjoyed. Based on what you mentioned, it seems like the mechanical properties of bone are depending on the exterior, and it seems like in vivo, the cell is changing the interior, and that doesn't kind of affecting the mechanical property. Then what is that meaning? Do you know? Do you have any idea what is that for?
Yeah, so what I think might be happening, this is all hypothesis, is that it's all about the crack initiation. Probably in the situation where we're doing 3-point bending, it's not so much about the propagation. These bones seem to fail very abruptly, but all about initiation. And since the initiation is going to be happening at the surface and not within the interior, we definitely see that it's on that tensile side of the bone. Probably what's happening is whatever modification is happening on that most outer surface of the bone is stopping that first initiation, but once it's there, it just propagates. So that's my theory as to why the interior may not matter as much as the exterior of the shell.
And are there any changes in maybe density in the exterior if something coming out?
Yeah, so that's actually another thing we're really interested in. And with micro CT, so microcomputed tomography, we did it at a pretty low resolution because we were looking at structure, but we can run it at a higher resolution and run it with phantoms to actually give us density variations. I don't know. The fact that we see these very dramatic changes in mineral to matrix content on the interior suggests to me that we probably do have a change in density, but in the process, I think micro CT will give us that answer. I'm surprised that in Raman we didn't see a change on the outside, but maybe micro CT plus Raman will give us that info.
Sounds good. Thank you.
Any other questions? Otherwise, I can ask my 101 question.
I have a question.
Thank you.
So yeah, Alex, it's such a nice talk. Yeah, I learned a lot about especially ketosis and bone health. So any ideas on what the mechanism of dissolution would be? Is it going to be controlled by sort of a surface reaction between the bone and the body fluid, or is it going to be dominant by the long range diffusion?
Sorry, I didn't get the question. Can you say that again?
Yeah, so my question is about the interaction of the body fluids with the bone material. So the reaction, we say it's a reaction. So is it going to be the overall dissolution process? Is it going to be dominated by, my question is really about the kinetics. So is it going to be dominated by the surface reaction, that is the availability of the dissolving species at close to the surface, or is it going to be the long range diffusion? Which is the dominant mechanism?
Yeah, so in these shorter term, I mean, the longest we're going here is 14 days, so it's not like a super long term. I definitely think it's a surface reaction. Due to the low porosity of bone, it definitely doesn't seem to be kinetically something that's happening quickly through the thickness. It definitely seems to be the surfaces and even the pulp on the interior. In the in vivo system, I didn't show this data, but it does appear that we have also changes at the interior surface where the bone marrow is. So body fluids in the bone marrow may also be having an effect. So I think it's mostly essentially the outside and the inside surface that are being affected, and the bulk is all cellular response.
Okay, so if the surface is going to be the dominant, the surface reaction is going to be the dominant mechanism, it also has to do with the shape of the bone. So that means depending on, because bone is essentially irregular shaped object to my eyes. So then depending on the surface of what is the surface area, there may be different dissolution rates. So is that something you observe on a bone?
Yeah, so this is actually a great question because the surface of bone and the geometry of bone tries to avoid any sharp edges, right? Bone is mechanical, so it doesn't want to ever form some sort of singularity where you're going to get a stress concentration. So when we think about things like, corrosion, stress corrosion cracking and things like that, the bone is already avoiding stress concentration. So we minimize those regions where we expect to get higher dissolution because of like stress corrosion cracking. But we did look at multiple different areas along the length of the bone. And we did see that the effects on the interior were more prominent in the middle of the bone than on the edges, what we call the epiphyses. And we're not sure why that is, other than maybe there's more cells in the middle than on the edges, or the porosity is allowing more flow. The other thing is there's lots and lots of studies about fluid flow in bone. And most of your bones, we think about compression, right? You're like, oh, I jump, I land on my bones, it's compressive. But we also have bending, like the bending moments on your femur are quite important. And it seems to be that bending really increases fluid flow essentially in the center. Just like 3-point bend, you're causing these huge variations every time you walk, so that acid may be getting to the cells more efficiently in the center of the bone than at the edges. And that could stimulate more cellular response. So the full geometry, plus you think about fluid dynamics, plus you think about cellular biology, it seems like such a simple tissue, but there's a lot of factors that go into this.
Yeah, thank you.
Yeah. But not stress concentrations. It does everything to avoid stress concentration.
Yeah. Thank you.
Any other questions, folks? I have a relatively simple question, I think. But if you're removing carbonate and calcium from a mineral, could that actually give rise to increased toughness, meaning, there's room for some diffusion or something to occur? It's giving rise to sodium to occupy different sites. I mean, I'm just, or is there some ion exchange going on that could give rise to some increase in toughness? I mean, of course, the matrix is maybe is getting enhanced, and that's the reason. But I was just thinking.
Yeah, so it's interesting because we do know that this calcium removal from lots of studies looking at these apatites geologically, because carbonated apatites do exist geologically. And so we see actually a lot of research on these crystals occur in, Earth and planetary sciences departments or geology departments. That removal of the calcium creates vacancies, and that those vacancies could then possibly have a mechanical effect. Now, we know from the study that we did with the mechanics of the crystals that when we were adding or removing carbonate from the crystals initially, that we were creating more vacancies. But the loss of calcium, like calcium and carbonate, seem to be very correlated. So usually what happens is you increase calcium, you decrease carbonate, and vice versa. But in this acidosis system, suddenly both of them are being removed. And that hasn't been understood from a charge balance perspective. If I'm taking out one carbonate, which it comes out as bicarb, so you have a -1, and I'm adding in, and I'm removing a calcium, which is +2, then I have to be taking out other things. So there's a lot of questions about, in these acidic systems, what does the ionic structure, the atomic organization actually look like? Where are my vacancies, and how does that affect this behavior? So maybe there is some work on it, and I don't have an answer as to whether those vacancies might affect mechanics.
Any other questions?
Well,
Everyone's looking at their bones.
Thank you.
Yeah, just based on my curiosity and doing a lot of protein work. So you already mentioned about the collagen, but with low pH, there is also possibility of some kind of denaturation of proteins. So maybe that will, anyway, those, the collagen is well cross-linked. So, but there are still room between cross-linked regions. So there's a possibility maybe collagen is somehow slightly denatured and probably give more flexibility or, the toughness. So that'll be a really interesting point to connect from the nanoscale to the, the higher scale. So.
Exactly. Yeah, I was really shocked by the toughness results, because usually when you think of bone, it's going to be a strength issue. And the fact that the toughness went up, usually you're like, oh, that's a protein question. When I worked in tendons, toughness was our big, our big changer. And I think you're right. I think we've ignored the collagen up till now, and we can't. So Margaret, my undergraduate student, is actually doing FTIR on the ex vivo bones to see, because that'll give us information about cross-linking and denaturation much more effectively than Raman ever does. So we're doing that, and then we're hopefully going to do some analysis for ages as well, which are, a different type of collagen cross-link, to see if we have reduced cross-linking or denaturation. Is probably my first guess. I mean, you take a tendon, you put it in these types of acid, the whole thing dissolves. Yeah. So yeah, I think there's a lot of interesting questions relative to the collagen, which I have been trying to ignore. And you're right, I can't ignore it.
Yep, sounds good.
Yeah. Any other questions? I can add, like, there are actually very few talks that directly impact us. Lots of talks you listen and you like the science, and at the end you're like, fine. But this talk is going to fundamentally change the way we think about our food. So,
and that's the advantage, yeah.
So on that note, let's thank Dr. Alix Deymier for a great talk. This was very, very refreshing. This is going to be one of the first biomaterials talks, but I think we're going to have a few more lined up. And this is going to be online, Alix. So this may come back to haunt you later, but be prepared for that.
I'll go back and be like, that's all wrong.
So if there's no other questions, I think let's thank Alix once again. And we've been promising all our speakers polos. Ankit's given up. We ordered these polos 1 year back, and we've yet to ship them. So you'll get eventually a polo with MateriAlZ logo on it. But great, and hope you can also log into other talks. I think you'll be on the listserv. I think Ankit's maintaining it. So And I know, have you met any of the faculty who are still logged in, who want to say hi to Alix? I know that BG was logged in. He probably left, and I think our new
It's
department head.
nerve-wracking giving talks to your old professors.
And I think
Had your classes.
a new department head was also logged in, Sammy, but he's not
Right.
here. But anyway, this was great. Thanks a lot, Alix. And any other comments from anyone else?
You can
I think
always
it was
track
great.
down my
Good to
email.
have.
if you guys
Good
have
to have a
Yeah.
biomaterials talk. Yeah, and I think I was able to connect to it much better than I do normally. So.
Perfect.
So Alix, and I think this opens up a lot of collaborative opportunities too. I think I'm sure Ankit's already thinking about some. Right? Microstructural evolution, surface reaction stuff.
It's a great topic to work on. Yeah, absolutely.
Okay.
Lots of
Great.
questions, lots of levels. But
Yes.
thank you, everyone. This was great. I appreciate you inviting me and all your great questions. Thanks very much.
Bye, Alix.
Thank you, Alex.
Bye, all. Bye, Christina. Bye, Ankit.
Bye, Krishna.